Good afternoon, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers, and I will be your technical host for today's webinar. I'm with the Forensic Technology Center of Excellence here at RTI. Today's topic is part of our SNP webinar series. The title for today's presentation is Predict Human Appearance from DNA, focusing on pigmentation. We will have two presenters today, but before we introduce our presenters, I would like to introduce the man behind the SNP series for us, Mr. Daniele Podini. Daniele, you might be muted. You can try unmuting your line.
Thank you, Josh, and thank you to all attendees. Welcome to the second webinar of the series titled All Things SNPs. Today's webinar will discuss SNPs that can be used to predict physical appearance, and we will be focusing on human pigmentation. We have two exceptional speakers, Dr. Manfred Kaiser and Dr. Susan Walsh, which will be formally introduced by Josh in a minute. And the format today, as Josh mentioned, will be slightly different from our last webinar. Dr. Kaiser will give his presentation, and he will be summarizing the existing genetic knowledge on human appearance. And after that, we will have a Q&A session for about 10 minutes. Then Dr. Susan Walsh will go into detail and discuss some of the tools that can be used by practitioners to predict eye and hair and skin color. And after that, we'll have a second Q&A session. So without further delay, let's begin. And back to you, Josh, to introduce our first speaker, Dr. Manfred Kaiser.
Thank you very much, Daniele. We'll go ahead and switch everyone's layout now to Dr. Kaiser's title slide. Dr. Kaiser's presentation today will be on genetics of human appearance and forensic applications. Dr. Manfred Kaiser is currently the Professor of Forensic Molecular Biology and Head of the Department of Genetic Identification at Erasmus University Medical Center in Rotterdam. He received his diploma in biology from the University of Leipzig in 1994, then his PhD in biology and genetics with summa cum laude from Humboldt University in Berlin in 1998, and his rehabilitation in genetics from the University of Leipzig in 2004. After his postdoctoral research at Pennsylvania State University, he was a staff scientist and later Heisenberg Fellow of the German Research Council at the Department of Evolutionary Genetics, Max Planck Institute for Evolutionary Anthropology in Leipzig. Without further ado, I would like to go ahead and hand things over to Dr. Manfred Kaiser, who will be sharing his screen momentarily and getting started with his presentation.
Okay, thank you very much, Josh. Do you see the slide? Is it okay now?
If you want to go ahead and hit the share screen button, that will bring that up for you. All right, perfect. We can see your slides.
Okay, great. Thank you very much, Josh. Thanks to everybody for joining. Thanks to Danielle for organizing this. It's a pleasure to be here to summarize what we currently know about the genetics of human appearance and how we can apply this to forensics. So as was said, I give a broader talk. I will cover pigmentation, but then we'll also a little bit going beyond pigmentation, more on the future aspects, and then we can discuss this if you want, and then afterwards, Susan Walsh will come back on more details on eye, hair, and skin color prediction from DNA. Yeah? So if you have more detailed questions, including technical questions, please hold, because very likely Susan will answer them, and if not, then she can answer that after her talk. Whereas the general question that you get after listening to my talk, we can happily discuss in the Q&A after my talk. So the first question, of course, then you may ask yourself or ask others is, why would you actually use appearance in forensics? And why would you want to predict appearance from DNA in forensics? Well, all being reminded, and we can see that many of you come from the practical side of forensic DNA testing, so you're all very much aware of that problem, that you can only identify people you already know, because forensic DNA profiling is a completely comparative endeavor. So you have to have the DNA profile of the person you want to identify. So if there is no match between the crime scene DNA profile and that from the criminal database or from the suspect you may have from other sites, and if you have no other information, then obviously it doesn't work with using DNA in the current way. So this is actually then how appearance prediction, and in the same sense also age prediction, ancestry prediction comes in, because if you can then do this, so if you can predict from DNA how a person looks like, how old a person is, where the ancestors of the person come from, obviously you can not only check if an eyewitness statement is correct, but also in those cases where there is no eyewitnesses, you can of course then give the police useful information that they can use in order to at least trace unknown perpetrators and find potential suspects. This, of course, can also be used in some special cases of missing person identification where you cannot use the current DNA profiling. So in that respect, this type of use of DNA is indeed very different to the current use of DNA, which actually, of course, is then the use in courtroom because it's the use of individual identification. This here is a use for investigation. So it's very unlikely to be in courtroom because you want to use it most likely very early on in the investigation, namely then when the police has no idea whom they shall be looking for, and then the DNA information on appearance but also age and ancestry may give them an idea. So the idea really is to reduce a rather large number of potential suspects by the outcome of appearance, age, and ancestry prediction from DNA. And then once you have that reduced group, namely all people that match the appearance, age, and ancestry information that you get from your crime scene DNA sample, then obviously you would still do DNA profiling because DNA profiling is individual specific, whereas appearance, age, and ancestry prediction for the time being is not individual specific. So why not? Well, obviously there is large information, large variation in appearance around the world, but at this moment we are not able to cover most of it, and we are particularly not able to cover the individual specific appearance information. What we can cover, as you will learn in the next 40 minutes or so, is appearance that is different between groups of people, but not yet between individuals. So unless that will change, and in principle it can only change if we are able to predict individual-specific facial appearance from DNA, this is a tool to reduce the number of potential suspects, whereas then individual identification is done subsequently via DNA profiling. So obviously there are people that look very much alike, and these are the monozygotic twins or the identical twins that share almost all of their DNA. And because we know that they share almost all of their DNA, and we also know that they are very, very similar in their appearance, in some periods of life, as you can see on these pictures here, they are basically indistinguishable based on appearance only, we know that appearance must be largely in our genes. The problem is that this information alone does not differentiate the possibility where appearance is in a small number of genes or appearance is coded in a large number of genes. So that, of course, is what you have to find out, namely by trying to map the genes that are coding for our appearance. And I will not go much into detail here how to do this, because I will rather show you later on what people have found by doing it, but just as a kind of educational wrap-up. So what this is indeed, it is about SNPs, and it is about the technique that people use for about 10 years or so, which is called genome-wide association study or association testing, where indeed they use SNP microarrays, so those technologies where you can do hundreds of thousands and nowadays more than 1 million of these SNPs in one analysis. And now people also start whole exome or whole genome sequencing data for this. And you simply ask for association, which basically is how often is a certain SNP allele found in people of a certain appearance. For instance, in the people that have blue eyes versus those that have brown eyes. So this gives you a probability of association, and because you do many SNPs, there are some multiple testing problems, so the convention is that you have to have a p-value that is smaller than 10 times or 5 times 10 to the minus 8. So it's a very small p-value, and you want to replicate those results in independent samples, and at the end you want a kind of functional proof that your associated SNP actually changes something, for instance, in a cell-based assay or even in an organism, for instance, in a mouse. So this is the whole Monty. So this is starting from knowing nothing, doing genome-wide association testing and subsequent analysis to then at the end establish that a certain SNP, a certain position in the genome, being involved in a certain gene, being involved in a certain gene function, makes a difference in appearance traits. So this is what people did for some appearance traits already, but by far not for all, and by far not yet enough and not yet complete enough for many appearance traits, as you see in a minute. But for those appearance traits where it has been done most complete, not to say completely complete, indeed are the 3 human pigmentation traits. And there is a reason, as Susan then will explain later on, why at this moment the 3 pigmentation traits are the only human appearance traits that we can predict from DNA with an accuracy that is high enough to be practically useful. So let's start with eye color. Obviously, there's large variation in eye color, as you see here on the left side of that slide. It depends a little bit where you come from within Europe, what eye color you have, because there are more blue-eyed people in the north and there are more brown-eyed people in the south. And if you then move away from Europe and nearby regions, basically everybody becomes brown eye. So non-brown eye color, indeed, is an indication of European ancestry, or at least partial European ancestry, because nowhere else in the world eye color variation has evolved, but in Europe. So people did find genes. People did find genes before the application of GWAS, but since the use of GWAS, people find more genes. And at some point, what we were asking is, so what if you take these SNPs that have been identified in those genome-wide association studies, but also in some other previous candidate gene studies, and you ask how useful are they to predict eye color? So what you see here is a representation of a prediction statistics that is also used in medical genetic prediction, and that statistic runs from 0.5, which is basically random prediction, so you can not even use your SNP, and you can just toss a coin, to 1, which means every time you predict correctly. And this was done in a couple of thousand Dutch Europeans, where of course we had eye color phenotypic information, and we had the SNP genotypes. And below you see the number of SNPs that are added to this prediction model, and if you know the right SNP, then what you see is you can predict blue and brown eye color quite accurately, because you get an AUC that is around 0.9, where again an AUC of 1 means that every time you predict, you're predicting correctly. If you then add SNPs to the model, you see that the accuracy increases, but you also see that it reaches a plateau. Why? Because if you add additional SNPs, these SNPs only have very small effects. And what you also see is that for non-blue, non-brown eye color, and these are not the greens because they're very rare, there's actually eyes that are blue and brown, this accuracy is much lower. So we now did this based on a test we developed that only includes 6 SNPs in more than 9,000 people, and you see that there is a very high prediction accuracy of about 0.95. Again, 1 means completely accurate for blue and brown eye color, but is not high enough for non-blue and non-brown eye color. So then if you look at this test and how sensitive it is, because we did a Swigdom-based validation study, you'll see it is quite sensitive. That has to do with the low number of SNPs and the use of that snapshot technology, which runs on a normal CE machine. So if you go down with DNA input at about 30 or so picogram, you still see complete profiles. If you go lower with input, you start to lose alleles, and that tells you that the amount of DNA is not enough for this type of test. So what we then did, and Susan will go into detail, is we developed a website that accesses our prediction tool where you put in the genotypes, you press a button, and then you're getting individual probabilities for blue, brown, and intermediate eye color based on the input genotypes. So that's a website that is publicly available, so everybody who has those genotypes can get those probabilities. So this is an example of 40 people from the department and across the floor, and those eyes are not ordered as you would order them by ordering the eyes according to eye color. The eye pictures are ordered as the test outcome orders them, with the highest probability for blue eyes here, and then the ordering of the color of the images goes with decreased blue eye probability, and with the largest probability for brown eye here, and then the ordering of the images goes with decreasing brown eye probability. So what you see is that all of these eyes here are blue, and that all of these eyes here are brown. There's one of the 40 that has a fairly high brown eye probability of 69%, but the eye color is not brown. And that actually indicates the error rate that you have with an AUC value across many thousands of people that is in the order of 0.95. And there are two individuals in this test set here, which is just to illustrate the power of the test, not to infer any statistical meaning, where you have different eye colors that have similar probabilities, where you would either conclude that the test actually is not conclusive, or you would conclude that the person is of mixed blue-brown eye color, which in this case would be a wrong conclusion, and in this case would be a right conclusion. Okay, so if you apply that test then to a worldwide sample, simply to see how it behaves in people of European but also non-European origin, what we see is that indeed it predicts brown eye color and only brown eye color in those areas where only brown eye color exists. And in those areas where you predict different eye colors, this indeed are European and nearby areas where also different eye colors exist. So let's move with hair color. Obviously, there is a correlation on the phenotypic level, and therefore it's not surprising that you also have a correlation on the genetic level, because many genes that are involved in eye color are also involved in hair color. So far, we only know one gene, which is this one here, MC1R, that is involved in hair color, also in skin color, but based on current evidence is not involved in eye color variation. So also there, people have identified genes. Again, most of these genes are overlapping. Some of the SNPs are even overlapping. But the number of genes and also the number of SNPs that you need for prediction is larger, which partly has to do with the prediction of red hair, which only is because of a single gene, and that's this MC1R gene, which is not involved in eye color. But there are many rare SNPs in that gene that all predict red hair color, so therefore in this test at least there are 11 SNPs just for red hair and another 11 for the other hair colors. So in that respect, a red hair color is an exception because it's basically a single gene trait, whereas all other hair colors are influenced by many more genes. So because of that, we can predict red hair most accurately from all hair colors, which of course is known for a long time already, because that gene is also known for quite a while to be involved in red hair. It's also quite accurate for black hair color, as you see here, and it's least accurate but still practically useful with these AUC values here for blonde and brown. And the reason for that is that there are some blonde people that actually get brown or dark blonde when they get older. And some of those are predicted correctly and others are not predicted correctly. And we don't understand why that is, and we also don't understand at the moment why some blonde kids stay blonde as adults, but others become dark blonde or brown as adults. So we also developed a test there, which we called HiRisplex, based on 24 SNPs for both eye and hair color prediction. So most of them are overlapping, but not all. And here you see the AUC values based on about 1,500 individuals that we have in our database at the moment. The test is also fairly sensitive. It has been SWIGDAM validated. The sensitivity threshold is with about 60 picograms, so it's a bit higher than the HiRisplex test, and that has to do with the number of SNPs being higher in this single snapshot assay. Again, the website allows to convert genotypes into probabilities for then the 3 eye color categories and the 4 hair color categories. So here are examples. Again, these images are not ordered according to color. They are ordered according to probability. On the left side with the largest probability for black hair, then largest probability for brown hair, highest probability for red hair, and highest probability for blonde hair. So those are indeed those blonde hair people that stayed blonde, but there are also those that predict highly blonde, were blonde as children, but then became darker, either dark blonde or brown, when they got older. So there, indeed, there is a problem, which at the moment, not only this test cannot solve, but no DNA test can solve, because we don't understand what the molecular basis for this age-dependent hair color change is. If you apply the test to worldwide samples, same story as with eye color, you predict black hair only in areas like East Asia, Africa, Native Americans, Oceania, where there is nothing else, at least in this sample, than black hair. So the only exception would be this part of the world here, New Guinea, Solomon Islands, where indeed there are black hair individuals, although those blonde hair individuals, have dark skin and dark eye color, as dark as their black hair fellows. We know that there is a mutation that is causing those the blonde hair, which actually then, in contrast to Europeans, does not cause any lighter eye and hair color. We know the gene is not the SNP is not is a different SNP in the same gene than involved in European hair color, which is not included in this test. So if you want to predict blonde hair in the Pacific, then this test is not useful. But that is actually an exception, because everywhere else in the world, except in Europe and nearby regions, we only have black hair color, which the test then correctly predicts. And here are examples, the more common examples like the blonde blue eyes, but also the more rare examples like the dark hair blue eyes, and also in the red hair color, the more, or both types, the blue eye red hair and the blonde and the brown eye red hair. And because we use different models and partly different SNPs, but particularly different prediction models, we can predict those eye and hair colors correctly in both type of combinations. The common combination of phenotype correlation, but also the not so common non-correlated phenotype situation. Okay, so what about skin color? One problem with skin color is that if you use genome-wide association testing, you will never be able to find all the genes. And that has to do with one prerequisite in GWAS, and that is that you have to use a genetically homogeneous population. If you do this, for instance, taking Europeans, obviously you don't have all skin color variation there. If you better do this then in South Asians, you have more skin color variation, but also not the maximum skin color variation, because the maximum skin color variation indeed is between Europeans, Asians, and Africans. But if you pool those, you're getting lots of false positives in your GWAS because there are other reasons for genetic differences than having different skin color. So that's the problem, and therefore GWAS is done within regions. They typically don't reveal the entire gene list. So we know genes, but the genetic knowledge, because of that worldwide distribution, is still more limited compared to eye and hair color. And therefore also the prediction that we can do at the moment is not so accurate. But that, as you will see in a minute, depends on which skin color category it is. So very recently, published this year, Susan and I and our collaborators then actually took data, took SNPs, produced data for 77 SNPs in more than 2,000 people from around the world, and then selected predictive markers, which is what you see here. So you also see that by adding SNPs, you increase prediction accuracy, again expressed by AUC, and at some point then you reach a plateau. So you reach a plateau not so quickly. So we then made a cutoff with 36 SNPs that we use for skin color prediction. And here you see the AUC values for 5 categories, because we were interested in not only predicting light, dark, intermediate, but actually to take it a step further and trying to predict different skin color also within Europeans, for instance, and therefore differentiate 3 light skin color categories and 2 dark skin color categories. As you see the AUC values, they are very high for dark to black, so for the darkest. They are quite high for dark. They're not so high for all 3 light skin color categories. Of course, if you would pool them to 1 light skin color category, the AUC value would be much higher. But we don't want to do this because we want to cover skin color variation and want to predict skin color differences, for instance, between Europeans, which does exist, but of course is smaller. And as you see here, the SNPs we have in that model are predicting the light colors less good than the dark colors. But it's a start, and we think it's a good start. There's a test which we are developing. A paper is currently submitted. Susan will tell you a bit more. I only show you 2 examples. This is a person that then is predicted for all 3 pigmentation traits. So we call this high-risk plex S for skin then, as you see here. So you get 84% probability for black hair in this person, 99% probability for brown eyes, and 94% probability for dark skin. And this is the person, so you can see if the predictions are correct. This is Lakshmi Chaitanya, a PhD student who now actually got her PhD, who worked on this and has developed that high-risk plex S test together with Susan and us. And here's a European example where you see someone that was blonde as a kid, but if you go for the highest probability rule here, you would see that the predicted hair color probability is rather brown than blonde. Also, you also see 40% blonde. Yeah. 69% blue eye probability and 66% pale skin, and you can see if it's correct or not. Okay, so I just learned before the talk that Danielle is planning an extra webinar on age prediction. I didn't know that before, so please allow me to just throw in a few slides on that. Why? Because as you have just seen already with hair color, it is important to have the age information because some of the appearance traits are age dependent. So the fact that hair color changes then, of course, makes it necessary to have the age information. Another example is male pattern baldness, which I come back in a minute, where you also, of course, have an appearance phenotype that in its manifestation depends on age. So therefore, you do want age, and as you will learn in a later webinar, you actually can do this. So a couple of years ago, we did develop a first molecular test for age by taking advantage of the fact that also the DNA sequence is the same, independent of age, because the DNA sequence doesn't change. What does change is a certain number of cells, because we lose these cells with increased age. And these are the T cells, and that's the reason why elderly people, they are more vulnerable to infectious diseases, because their T cell-based immune response doesn't work, because they have a small number of T cells. Because what happens between birth and our death is that the T cells actually get converted into fat cells. So with a little trick, then, we targeted DNA that only exists in T cells. And if you do this and you predict age based on that quantitative DNA test, and you compare that with the observed age, you see that you have a very high correlation. So it's an R-square value of above 0.3. It comes with a standard error of plus minus 8 to 9 years. So it's not good to predict age per year, by the year. But as you see here, if you just want to know if a person is rather 20, rather 40, rather 60, or rather 80, this single test is already quite informative. So of course, if you want to improve this, then indeed you either have to move away from DNA and you go to RNA, because it's the gene function and not the gene sequence itself that can change with age. And here you see expression analysis, which is actually gene function analysis of young and old people. And if you then order them according to these gene expression data, you see that the people order according to age, with a single exception. You can also do this with DNA, and that, I think, is then what the next webinar, or one of the next webinars, will be about. But you don't use SNPs. You use a modification of the DNA, which is called DNA methylation, and you will learn more about that in a later webinar. Why? Because this DNA methylation is also involved in the regulation of the genes, so therefore this information here is related. And if you do this genome-wide and you find markers that are age informative and you apply them to young and old people, you see that they almost perfectly differentiate the young and the old. So if you then take selected markers and you ask how indicative of age are they, you get quite high R-squared values. And that's not only what we found, but that's what a couple of groups have found. So here you see several papers and models based on DNA methylations to predict age, and this is the error these models come up with. So that's mean absolute deviation, so it's plus minus 2, 3, 4, 5 years about. Yeah, that's about where you are with the accuracy of DNA-based age prediction, and you will hear more about that in a later webinar. So where are we before I come to the things that aren't possible at the moment? So as you were hearing, not only with those tools that we developed, but also with tools other people have developed. Susan, I think, will cover some of that. You can predict categorical, and that means groups of eye color, groups of hair color, groups of skin color, and age. So for that, people have developed markers, people have developed prediction models where you put in the genotypes and you get out the phenotypic information. And people also have developed lab tools where you can then get those markers analyzed in DNA that is of low quality and quantity, such as those you get from crime scene samples. Yeah, don't get confused, because if you have enough good quality DNA, and you can therefore analyze a large number of SNPs, partly you can increase the accuracy, particularly when it comes to age. But these tools, these arrays, DNA methylation arrays or SNP arrays, they typically don't work in forensic DNA. So everything else we cannot yet do. And the reason is that we don't understand the genetics well enough. And here you see a list of things that you may think of when thinking about other appearance traits. I will show you a few examples of those where the research stands at the moment, also to explain why we cannot yet use this for prediction in forensics. And so one example is eye color. Obviously, the idea that eye color come in three groups is not very realistic because there isn't only three groups. Eye color is a continuum from blue to brown, and as you see here, if you then label blue, brown, intermediate in such a digital color space, you see that the blues are here, the browns are there, and the intermediates are here, but of course you don't see three discrete groups. Why? Because eye color is not a categorical trait. It has been simplified as a categorical trait in order to make it easier to find the genes. So if you then quantify eye color, for instance, from digital images, which we did here, and you use that in a GWAS to try to find genes, indeed in this study, in a couple of thousand Europeans, you do find 3 more genes, and that can be explained by the fact that you use more phenotypic information compared to the previous studies where categorical eye color was used. So this is what people are nowadays doing. What they also, of course, do is to find more genes, simply increasing sample size in the GWAS and keeping with the limited categorical information, for instance, because it is easier to get such information, for instance, from questionnaires than to collect images and quantify it with some computer methods. We also did this for skin color. We also found some more genes here, at least in a candidate gene study, and actually did not find new genes when we did the genome-wide association study on continuous skin color in almost 20,000 Europeans. We did find genes that were genome-wide significant, but they're not new anymore. So this is an example where if you move to more phenotypic information because you use continuous information, at least in this study we did not find new genes. Why? Most likely because all of these new genes that are there, we know they are there, but we cannot find them with statistical trust. They have such a small effect that you cannot identify them trustworthy in a number of 17,500 people. So this can be illustrated much more convincingly when we move to another trait, which is body height, where we know most of body height information is in the genes. How do we know this? Because for that people use twin studies where you can estimate how much of a trait is heritable, so is inherited to the next generation, and therefore it must be in the genes. Again, you only know by that more than 80% of height variation is heritable. You don't know if this then is 1 gene or 1,000 genes, unless you start doing genome-wide association testing, which people did. And here actually the knowledge about the genetics of body height variation, at least when it comes to normal body height variation, not to the extreme small or large body height variation, this was only produced after people applied genome-wide association testing. So these are all GWAS studies that actually allowed people to understand the genetics of body height. What's the problem? Well, the problem again is in the small effect size. So here you see a study we did in 17,000 Europeans. We did find a new gene. Here you see the genes, or there's actually more than one in that area, lower than 5 times 10 to the minus 8 association p-value. So we trust this, and we could also replicate this. The problem is here. So with that SNP that shows a nice and trustworthy genome-wide association that has been replicated, the effect on body height is 4 millimeters. And I tell you that this is a large effect. So most of these body height SNPs have an even smaller effect. So therefore, if you increase sample size, 130,000 people, you do find more genes, 180 genes, but unfortunately, because of the small effect of these genes, they only explain 10% of the height variation, where 80% should be in the genes. If you then further increase sample size to find more genes, you do find them, 700 we know at the moment, that are below the significant threshold, so we believe in them. How much do they explain of the height variation? 16%. 80% should be there. So that indicates the big problem of common complex traits, which all of our appearance traits are, because the phenotypic information is distributed across many genes. And basically all of those genes have similarly small effects. And that differentiates appearance traits that are not involved in pigmentation from those that are pigmentation traits, because there we have at least one or two genes that have a larger effect and many others that have a small effect. And that's the reason why we can predict pigmentation traits at the moment, whereas for all the other appearance traits, it seems that all the genes have rather small effects. And that makes it difficult to find it. But it also then provides a challenge for DNA breast prediction, because as you see here, the number of SNPs that predict a trait become very large. And then you get an issue with what technology you should use to analyze them in the low quality, low quantity DNA you get from a crime scene sample. So we thought about a shortcut, which doesn't really work, but gives you a bit of an idea how that may develop, because we try to predict extremely tall people. If you do this with 180 or so SNPs, or a subset of those, then you end up with an AUC that is basically in the middle between random prediction and completely accurate prediction. So it's not yet there in terms of application. Same applies for hair loss. Hair loss in men, that's baldness. I typically show this here just to illustrate that if the men amongst you listening now and seeing these slides so far look at their father, if they want to see whether or not they get bald with age, that's not what you should do. Why? Because although it is a trait that has many genes, also on other chromosomes. The gene on the X chromosome actually has the largest effect. And of course, as a man, you get your X chromosome from your mother. So you should actually look at your maternal grandfather if you want to know whether or not you're getting bald in 20 or 30 years. Can you predict it? Well, not yet. That was a study using 11 SNPs. Nowadays, we know more. The AUC values you get, again, are around 0.7, which is basically between random and completely accurate. There was a recent paper that has revealed hundreds of new SNPs being involved in hair loss from hundreds of genes. How much do they increase prediction accuracy? Yeah, they do increase it, as you see here. So the extreme form, severe hair loss versus no hair loss, which compares to what we had seen before, AUC of 0.78. Do you want to use this? Well, I think this is where it starts to become useful, because, of course, that's an average across a large number of individuals, and that includes many individuals that have, on the individual level, a high probability. But of course, there are also some that have a low probability. What about facial shape? There may be another webinar, Danielle said, on face genetics per se. Just to give you a bit of a glimpse about that. Obviously, this is very complex, as people would only expect, which means it's very hard to find genes. People have found genes, typically by GWAS. There are a couple of those studies out there. Here I show you one example, where we mapped facial variation via MRI images and then also used photos to replicate. We did find genes, 5 genes. One actually overlapped with a parallel study that was the only one gene people found in a study using children. It was a bit surprising, although we know that facial appearance doesn't change completely with age. Typically, people can recognize someone from a children's image, and that's what this reflects. But of course, there's also a change there. So one of the 5 genes actually in adults overlaps with that found in children. So what is the effect size? As you may already expect, it is small, and that's the problem. So here is the gene that had the largest effect of those 5. It actually influences eye-to-eye distance. And this is the effect size you get when you're heterozygote for that SNP or when you're homozygote. So it's 1 or 2 millimeters. So what's the eye-to-eye variation, at least in this sample set? It is about 20 millimeters. So those 20 millimeters variation at the moment with this one gene and one SNP, we can explain 1 or 2. So that shows you how complex it is and how much work is still needed to be done to understand more of the genetics of facial variation, but also then to use this later on for prediction. So I finished with an example, just to summarize that. So what's the problem? The problem is that with current DNA profiling, you can only identify someone you know already because you need the DNA profile. With forensic DNA phenotyping, at the moment, we can do eye color, hair color, skin color, age, at least approximately, also ancestry, and you will also hear something about ancestry prediction in a later webinar. And you can apply that already to forensic cases, as people do. There are labs that do this. Of course, you have to be in a country where this is allowed. In some countries, it's not. But you can also use this here in missing person identification. And there, actually, the legal constraint on using this in many countries isn't there because the forensic DNA laws, they apply to criminal cases. And I'll show you one of those examples. Not that there was a legal constraint there here, because as you see here, this person is 500 years, more than 500 years dead. So this is King Richard III, the last king of England who died in the battlefield. And Shakespeare, of course, wrote about him, as you all know. So that's him. He was identified underneath a parking lot in Leicester City. So you can read about that entire fascinating story in a paper published in Nature Communication. So there was multiple evidence to identify him. One was genetics, and that was anti-DNA genetics, because they were able to identify a maternal relative of King Richard III. And if they then did a whole genome and anti-DNA analysis, they found a perfect match between bone DNA and that person who, based on family records, is related with King Richard III on the maternal side. Those that followed the press about that probably saw that the other way to do this, via the paternal line using Y chromosome DNA, did not work out. So there was no match there, which basically means there was an illegitimate paternity somewhere here in that line. Anyway, what we were actually involved in this study is predicting eye and hair color of King Richard III. And what we predicted is 96% probability for blue eyes, 77% probability for blonde hair. These are people that have such blue eye and blonde hair probabilities. And as you would expect, they are blue-eyed phenotypically, and they can be blonde, or in case they did darken, can also be dark blonde or even brown. So the conclusion would be that King Richard III likely had blue eyes and blonde, or in case he did darken with age, dark blonde and brown hair. If you look at portraits, you see him with all types of eye and hair color. Apparently there is no portrait that shows him while he was still alive. This one here is the one that was painted most recently after his death, and indeed it does show him with light eyes and with brownish, light brown hair. So indeed it would agree with the DNA result. However, if you look at other paintings done later, you see him with black hair and dark eyes, and there probably was then the painter's imagination and not the reality anymore. So with that, I can finish. If you want to read more about that, there's a review paper published in 2015 that actually summarizes all that knowledge much more in detail. And that is also partly covered in a larger overview article in Nature Reviews Genetics that shows how you can improve human forensics true advantage in genetics, genomics, and molecular biology, not only on appearance prediction, but also on many other areas. So with that, I'm done, and I guess there is time for questions now.
All right, thank you very much, Dr. Kaiser. We do have a question. It's actually a two-part question that came in from Sushana, so we'll go over the first part. Her first question is, have you also researched people with black hair that eventually goes gray?
Yeah, so losing hair color and then becoming gray or white, of course, is an interesting trait. It also shows that besides the darkening, for instance, from blonde to brown, there is another age-dependent mechanism with hair color, which actually is particular then and is not happening really with eye and skin color, and that's, of course, losing the hair color. So there's one genome-wide association study that looked into this. They found only one gene that certainly has to do with a limited statistical power in that study because of sample size. And that actually is a gene that we know is also involved in pigmentation. So it's not quite clear why this showed up in actually losing pigmentation, but that is the current evidence. So we expect it to be largely genetic. Why? Because there are some people that actually gray very early in life, well, not very, but early in life, so when you are 30 or 40 or so, and that typically runs in families, so we would expect this to be genetic, but people haven't done the work yet to find the genes.
Okay, and her second part, the second question, is how did you determine that we should look at our maternal grandfathers to note if a male is going bald?
Because the gene that has the largest effect is on the X chromosome. And a man, and this only applies to men because this is male pattern baldness, they receive their X chromosome from their mothers because they receive their Y chromosome from their fathers. And therefore, it is the father of the mother, because that is then the male on the X chromosome line that allows an idea about baldness. It is not a complete idea because there are many more genes, and all of those other genes are not on the X chromosome, so you also get them from your father then. But they have a smaller effect.
Okay, and just a follow-up question to that. Should we, for female pattern baldness, be looking in the same area?
Yeah, so female pattern baldness is a different trait. So those are suffering, those women that are suffering from that, they know this because you're hardly getting bald. What happens is that the hair becomes thinner, basically equally across the head, which is another trait. The genetics, at the moment, much less is done on female hair loss. There is some overlap in the genes between female and male hair loss, so there seems to be some genetic relationship, but it certainly is not a complete genetic relationship. So I guess if people do more work on female pattern baldness, that will then determine to what degree you can also predict that. It is a much less frequent trait among females than it is among males.
Let me jump in for a second. First of all, thank you, Manfred. That was a great presentation. I do have a question as well. You were talking about individuals that change colors from blonde to brown, and that right now we don't know much about that. But do we know whether this is an inheritable trait, meaning two identical brothers will change hair, both of them, because of this?
I don't know if people looked into quantifying heritability for that. Of course, for that you need more information than just hair color, because you need to know information about hair color as child and as adult. I would expect it to be heritable. I mean, in my own family, two brothers were blonde as kids and are not blonde anymore. So I would assume it is largely heritable, but people haven't found yet the basis of it.
Yeah, because I was wondering whether it could be somewhat environmental. And the fact that two brothers change color, it could be because they're exposed to the same environment. Maybe also looking at methylation might provide some insight on that.
Oh yeah, that's for sure, right? Because I mean, the DNA is one thing, and that should determine a color. But then once the DNA is there, either you have a regulator that also is a gene that then makes the change, or you have some other molecular substance, which of course should still be coded in the DNA somewhere, that then provides the change. Could be epigenetics, it could be something else. At the moment, we don't know.
Okay, well, thank you.
All right, thank you very much, Dr. Kaiser. Do you have any parting words that you would like to give our audience before we move on?
Not really. I mean, if people don't have other questions, then I'm happy to have Susan continuing.
All right, thank you so much, Dr. Kaiser. We definitely appreciate your presentation today. We are going to go ahead and move on to our second speaker for today's webinar. Our second speaker today is Dr. Susan Walsh. She will be presenting on forensic DNA phenotyping, a pigmentation profile from DNA. Dr. Susan Walsh received her Bachelor's of Science with honors in biochemistry from the University of College Cork in Ireland. She then pursued her Master's of Science in DNA profiling at the University of Central Lancashire in the UK and worked as a research assistant at the Medical Genetics Department of the University of Sydney, Australia. In 2013, she gained her PhD in forensic genetics from Erasmus University in Rotterdam and followed this by her postdoctoral work at Yale University in New Haven, Connecticut. In 2014, she became the Assistant Professor of Biology in the Forensic and Investigative Sciences program at Indiana University, Purdue University in Indianapolis, Indiana. And without further ado, we're going to switch everyone's layout now. And Dr. Susan Walsh, if you want to go ahead and share your screen, you can start with your presentation.
Great, thanks Josh, and thank you everyone for coming in and listening to us give this talk this morning. As Manfred said, we're going to focus on pigmentation because that is the most progressive trait at the moment for prediction from DNA. What's possible? Well, as you all know, it's probably one of the main characteristics that you describe to someone as a physical appearance trait. You discuss as an eyewitness, you would say, well, what was their hair, eye, or skin color? So basically, it's one of the most obvious traits that we could focus on during our research over the last 10 years. And the phenotyping potential of pigment is immense. But as Manfred said, it is quite category dependent. We are not yet at the level of individual pigmentation prediction, but certainly we are capable of predicting categories. So first, let's begin with eye color. It's one of the first physical appearance traits to be studied for prediction from DNA, and one of the most successful. Finding the genes involved in eye color began, as I said, about 10 years ago with general genome-wide association studies and also model studies from animals. We began to understand the pigmentation pathway overall. And basically, genome-wide association studies really led the way because they helped us unearth SNPs that are highly associated with a certain trait. And in 2008, there seemed to be a surge in this research as we began to understand one of the main predictors or one of the main associated SNPs with eye color. This was followed then in 2009 and on by eye color prediction attempts. So basically, you can have an associated SNP with eye color, but you also have to assess is it useful for prediction, because these are two different questions. Association, yes, these can be found, they're markers. Large genome-wide associations enable us to find these markers, but then you have to assess, well, is it useful? Can we actually predict eye color using this marker? And in 2009, Manfred and colleagues actually looked at this. They looked at every eye color associated marker, and they assessed their ability to predict. So this systematic study really began to rationalize, to basically rank how good these markers were. So from there, Fan Liu, as part of Manfred's group, he assessed 37 SNPs from literature from 8 genes in over 6,000 samples from the Netherlands. And he assessed their contribution towards prediction of eye color based on 3 categories of blue, brown, and intermediate. Upon ranking these, there were several markers that really popped up as being highly predictive for this trait. In essence, 6 were key predictors that gave a very high accuracy of over 90% for eye color prediction. So 6 key SNPs from 6 genes were found to be very useful for eye color prediction. Always you have to have a model, and a model in essence is how we are able to control the prediction. Basically, if you have a large set of individuals that you can assess their genotype compositions, you can basically give a probability effect based on those markers. And this is what Fan did. He basically assessed the individuals within his training set of over 3,800 individuals from the Netherlands. Obviously, it is in Europe, and then there was a large degree of variation of blue, brown, and intermediate from that country. And as Manfred said, eye color variation is restricted to Europe and surrounding areas. And from assessing his model and testing it on over 2,300 individuals, separate individuals that were not involved in the training of the model, he found that just using these 6 SNPs from 6 regions within our DNA, we could actually predict blue and brown accuracy over 90%. And that's a very good trait to be able to predict, because you've got 50% level is the toss of a coin, 100% is perfect prediction. And we were close to that 100% prediction with these 6 markers. From there, always in forensics you need a tool. And at the end of the day, we wanted to create a system that you could basically have a model and an assay and generate this information that you can have a final result then from looking at an unknown individual's DNA sample. As Manfred said, this is all very good work for intelligence. We will never bring this aspect of research into the courtroom. This will only be used in intelligence. That's where we saw this research going, because STR profiling is really the gold standard of DNA profiling, and this will never be replaced. But as he pointed out, sometimes there are no matches, and to factor in an intelligence tool is where we saw this system going. So IrisPlex was created. It was a 6 SNP assay, which I'll talk about in the next slide. But basically, these 6 genes give their contribution towards eye color. As I said, back in 2008, we found that HERC2's RS129-13832 give a really high association factor with eye color. And through the systematic study, we found it was the most predictive marker for eye color. And so we asked the question why, and Manfred and his group actually tried to find the causal reason for this marker. And they did find that it was although intronic marker, so it does not code for a particular protein, it is an enhancer of the gene next door. So it is a long-range enhancer, and a particular genotype will either upregulate or downregulate the production of melanin in OCA2. So we try to assess first an association, secondly a prediction, and it's always good to find out the actual fundamental side of what's happening at the genomic level. So from this, many models have been created that incorporate this major causal predictor gene. And as you can see from the hypothesized scenario, it really is a very good indicator of blue and brown, with additional contributions from the additional 5 SNPs within this prediction model to either give a more darker brown eye color or more lighter blue eye color. Understanding the model, it is very easy to understand and incorporate and use for DNA profiling intelligence work. Most of our papers include a supplementary table that have this interactive macro, which you can see here. But we also have an online web tool, which I'll discuss later on, that is free to go on to genotype your results in the assay and in the lab, and then hop online and actually generate your own probability values. For eye color, we use the highest probability as the most probable result. So when you're talking about statistics and probabilities, all values sum to 1. But the highest probability we give is our probable result. And just as an example, I'll show here, we take the highest probability value for blue, in this case on the left. The individual was predicted as being 94% blue eye color, our probability of 0.94. And on the right, we have an individual with brown eyes with a brown eye probability category of 0.76. So Irisplex, as I said, we began with the model assessing a systematic study of what contributes the most to eye color prediction. We then progressed with developing the assay. Of course, we want to test the assay more. So from 2011 and 2012, we did additional performance testing on more individuals, more worldwide individuals. We also did a SWIGDM assay developmental validation. That's the scientific working group on DNA analysis methods, which I'm sure you're all well aware of. And this is just to show that the assay can be run by anyone in any lab using the equipment that is freely available to you now. So everybody uses, to my knowledge, capillary electrophoresis machines for STR profiling. Well, basically you can do the same thing here with these assays. It is a very easy to follow protocol, which is published. And also I'm available for questions if someone does have questions on the assay portion, to generate these genotypes and then to use the freely available web tools. So in essence, you could actually run eye color, hair color, and skin color prediction in your own labs tomorrow. So we then assessed an additional performance on 3,000 individuals from a scattering of Europe, just to show, as Manfred said, the difference in prediction. Northern Europeans having more blue eye color, Southern Europeans having more brown eye color. And we also showed that the sensitivity of the genetic assay was extremely sensitive, down to 30 picograms. So in cases, as many of you are DNA analysts, in cases where you find that you have DNA but it's extremely low or extremely degraded, you can still run these assays. You will still get a result from these assays. And that is extremely helpful, and it may point you in the right direction for police investigation. In 2014, we always like to improve our models, and as we generate more data and more individuals, we like to enhance our models. That's why having it online as a web tool is the best resource, because we can add to the prediction models and make them better. So by including the large variation within Europe, we were able to rejig the model, and now eye color for blue and brown accuracy levels are nearly reaching 95%. Intermediate eye color we are still working on. It is mostly down to the fact that eye color, as Manfred said, is not categorical. It is a continuous trait. And when we tend to put non-blue or non-brown eye colors into one category, it gets termed intermediate. And so genetically you are not actually predicting intermediate, because there is it's an amalgamation of many colors, hence the prediction is lower. But on the blue and brown angle, we are extremely good at predicting eye color. That is the website there, which I'll focus on in a minute and just give you a few examples. Very important, we want to make sure that it is not reliant on ancestry. We want to be able to predict color. It is the pigmentation palette that we are interested in. That way we are not confounded by ancestry or admixture results, and we don't rely on anything but pigment. Think of a painter and its color palette. That's what we want to predict using genetics. And we did test this, as Manfred said. Although we didn't have the phenotypes for these individuals, it does conform to general knowledge of the eye color distribution around the world. We are not the only group, and I always like to point out that there are other groups that have prediction models. There are other groups that have done research. My feeling is that our Irisplex and Hirisplex and Hirisplex S along with the snipper suite, are probably the most up-to-date and most reliable predictor models. It really is dependent on you as a user, which one you prefer. We like to use probability-based prediction for our models, and the snipper suite tends to use a Bayesian likelihood ratio type approach. Yet again, HERC2 139 13832 marker, the causal SNP that we found for eye color prediction, is in these prediction models as well. It is actually composed of 6 Irisplex SNPs with an additional HERC2 OCA2 complex. And they have several models available online for their snipper suite. Although they have additional models and additional markers, where they lack on is the number of individuals within their training set. So I've always said, and it's just general knowledge, the more individuals with the more variation you have in your training set, the better your prediction model. And we're approaching over 9,000 individuals in our prediction model. However, this model is just about 400 individuals. Implementation is easy. You can check out the website as well if you want to check out this snipper eye color classifier. On to pigmentation for hair color. Naturally, the progression of research led from eye color to hair color and the integration of a tool that can do 2 in 1. But actually, hair color was earlier in research and its ability to predict, but red hair color in particular. Some of the variants and some of the genes you will see are familiar because it's all part of the pigmentation pathway. From 95, unearthing red hair variants, all the ways to 2010, there was a surge in trying to understand hair color variation and its association. Previous work in hair color prediction, as I said, it began a lot earlier than eye color, but solely for red hair. So they were not able to actually predict brown, blonde, and black hair variations. It was mostly down to the MC1R mutations that they were able to predict red hair color. So their prediction models consisted of, did you have red hair or did you not? The prediction of all color categories, as I said, proceeded, and then in 2011, back again, we tried to assess with our colleagues in Poland and Manfred's group. We tried to assess in a systematic way through literature and everything that was ever associated with markers and hair color. Could we rank these associated SNPs? Could we provide a most effective small number of markers that can give you a high prediction accuracy? So through a model-based prediction approach, we found that there were 13 predictive DNA variants from 11 genes that could actually provide a high level of prediction accuracy for hair color. These are consisting of 11 single SNPs from the MC1R gene, which is associated with red hair prediction, and 2 combined sets of variants, to a total of 22 markers. So 22 markers enabled us to have a high prediction accuracy for hair color in the range of 80 to 93% for 4 categories of blonde, brown, black, and red. As I said, we like to make tools. We like to provide an easy source for forensic practitioners to actually generate the genotype data that they need in a reliable fashion, and use the prediction model of their choice, whether it be the Irisplex online model, as I'll show you later, or the snipper tool that uses the likelihood ratio approach. In 2013 and 2014, we ran through SWGDM validation again, and we assessed the model, improved the website and the web tool. For the assay, the SWGDM validated assay, we are extremely sensitive again, down to 60 picograms input of DNA to generate these 24 markers that are needed for eye and hair color prediction. So Irisplex was eye color alone. Irisplex is eye and hair color together. 22 variants are for hair, 6 variants are for eye color, and 4 are overlapping. As you can see, it's a very easy assay. It looks similar to an STR profile with the same color peaks. These are all based on genotypes, and when you're more familiar with the snapshot technology, you'll be able to recognize that each of the colors is a certain base nucleotide. The Swigdom validation occurred in 2014 of the Highersplex model, and we actually launched the website that we will focus on towards the end of this talk. We also enhanced the prediction model. As I said, we like to improve continuously to make our models better. And now for AUC values, accuracy values based on the area under the receiver operating curve, which is basically like a test for 50% toss of a coin, 100% real accuracy. We range from about 0.75 in brown hair to 92% in red hair color. Hair color is a little bit more complex than eye color. With eye color, we tended to use the highest probability as the most probable prediction result. With hair color, because of the more variation that you can observe, especially with the degree of shade. You know, I can tell you that you have brown hair color, but is that light brown or dark brown? And that's certainly a different color visually. And so we try to include that in our prediction models. Hence, they're a little bit more complicated and need a prediction guide, and that's what we include in our publications. So we do use the minor allele input from all 24 variants. 22 of these are required for categorical hair prediction. As I told you, the range is from about 75% of brown accuracy hair prediction, and that also includes that error that we factored in for blonde hair prediction, which Manfred's talked about. That blonde-haired individuals sometimes appear genetically as blonde, but phenotypically as brown if they are one of the few individuals that had that hair color dependent change as they aged. It doesn't happen in all blonde-haired individuals, but we try to incorporate this into our accuracy measures. We also included the shade or light-dark prediction of the hair just to give that added prediction bonus for hair color. And just as a visual example, here are a couple of individuals and what the actual output is from our prediction models. So we receive a category, whether it be black, brown, red, or blonde, and the highest probability, we begin with that. And then we add on the shade element to the prediction. So if I zoom in a little bit further here, you can see that this individual, if we are following our guide, which I'll talk about in the next slide, has a very high probability of blonde as a category, with an extremely high probability of light. So we can then infer that this individual's prediction would have a light blonde profile. As I said, we needed a prediction guide to enhance this result because you're talking about an increase in categories and the light shade category as well, as it being a more complex trait than eye color. So if you follow the guide, we have it stated that 70 or 0.7 probability is our cutoff range, where we tend to follow yes or no additional questions, and then the final prediction is regarded then as the most probable hair color result when you incorporate both the category and the light shade. We also wanted to make sure that ancestry was not a factor here because especially in the United States, you have a large proportion of individuals who are admixed from all over the world. And so we did not want to rely on ancestry as a factor in our prediction models. We wanted to rely solely on the color palette. And so we tested this on HDP, a worldwide panel. Obviously, we don't have the phenotype for this, but the results actually do conform to general knowledge of hair color variation around the world. Manfred did talk more on the Melanesian portion of hair color and the blonde individuals that are there. It is quite rare to have that variant and rare to have that prediction, and it is in such a low number that we did not incorporate this into our model. We incorporated general variation with more of a European variation for our hair color markers. Snipper also has a tool for hair color prediction. They use many, if not all, of the markers that we use for red hair, and also additional markers for brown, blonde, and black hair colors. They produced this tool in 2015, and it's on their website. However, there's no great improvement of AUC accuracies. They also have a smaller training set and a smaller test set. 7 overlap with Highersplex, many of them, as I said, MC1R variants. You can find this tool online at their SNPR website, which is available down there at the bottom. But there was no great improvement in prediction. So basically, it's up to you whether you want to use probability prediction using Hiersplex or likelihood ratio based Bayesian prediction using SNPR. But do factor in that there is a high degree of variation in the number of individuals used for the training models. Right now we have over 1,600 individuals for hair color prediction using Hiersplex, whereas SNPR tool has less than 500 individuals. Moving on to pigmentation, as Manfred indicated, for skin color it is more difficult. Genome-wide association studies for eye and hair color really pushed research boundaries and unearthed many associated traits or associated markers with this trait. Because they could focus on Europeans and not be affected by population stratification and using different individuals in genome-wide association study, so different populations. This is not the best approach for genome-wide association studies, having a worldwide set, because rather than getting the marker of interest or the marker associated with the trait, you tend to get markers that are associated with ancestry. And so you want to avoid that. So with skin color, many of the early studies actually focused on positive selection and the more evolutionary aspect of how we evolved our skin color. And this began in 2006, 2007. And they actually unearthed many genes in the pigmentation pathway related to skin color based on positive selection. So still we weren't at the level of genome-wide association testing yet. Progression then occurred, but only at the singular population level. And as you can understand, the variation in skin color within one population is not very extreme. Okay, you have the subtle variation within Europeans of the more northern Europeans having paler skin and the more southern Europeans of having darker skin, but they're still quite pale versus African darker skin. And so many of the genome-wide association studies were limited to singular populations, which makes it very difficult to find markers that can be used on a more global predictive model. Variation in skin color, there was a lot of European studies done, and most recently there was an African study done. And many of the markers and you can see here, are from genes that are well known, that we use for either eye or hair color. Many of them overlap, and specifically, and what's interesting is that many can be found specific to those populations. So further testing and further assessment would really factor in or basically answer the question, well, can that gene or can that variant actually be beneficial for prediction on a worldwide scale? Because it might just be specific for that population's variation alone. SNPR actually produced a tool for this in 2014. This can be found on their website as well. They used 59 SNPs in a systematic study in an attempt to assess, well, what are the most predictive markers for skin color from Europeans and non-Europeans, and they had several populations worth investigating. They did use skin reflectance measures and a questionnaire-based phenotyping to assess their individuals. And they used very simple categories of white, intermediate, and black classification. From the 59 SNPs, they found that 29 were associated with skin pigmentation. Those 59 SNPs were general pigmentation markers from literature, many of them found in eye color and hair color prediction models. And so when they assessed them for skin color prediction, they found that 29 were significant, but that 10 were needed for their prediction model to give high, good enough accuracy levels. High differentiation between white versus the rest was found in their study, but there was a high degree of overlap between intermediate individuals and black individuals. So although their prediction model is very good at predicting white or pale individuals, they had more difficulty differentiating between intermediate or more darker individuals and black individuals. So they had their difficulties within the model. Also, their training set was very low. They had 118 individuals as their training set, using 3 populations of white, intermediate, and black based on skin reflectance measures. So these numbers can certainly be improved. But it was one of the first skin color models to be used, and it is found on their website if you're interested in looking at skin color prediction with this classifier. Their accuracies, as I said, AUC can be seen here as quite high, but this is actually on the full set of their individuals. If you break down their predictions, there was actually 62% only correct for intermediate and quite low, only 30% correct calls in black individuals. And so overall about 20% were misclassifications. So certainly there was areas to improve on. That was tested on 167 individuals from very limited populations, and as you know, skin color variation around the world is so different, and due to the admixture of many individuals within the United States, it is essential to be predicting more accurate skin color and more global skin color predictive measures. In 2017, this year, Manfred and I then investigated more markers and more individuals, more global individuals for their skin color association effect. We looked at 77 markers from literature, from genome-wide association studies, to really assess in a systematic study, well, on a global level, what are the most beneficial markers to be used for skin color prediction. And this was published in Human Genetics most recently. We assessed our individuals based on questionnaire data and a dermatologist. So we had a dermatologist assess either physically within our Polish cohort or via questionnaire and physical photos of the individuals. We had a Fitzpatrick scale assessment. So Fitzpatrick scale is a dermatological scale from the 80s that bases skin color variation on UV exposure. So you have individuals that are deemed white that would have a skin color category of 1 to 4, brown and black. And this was the assessment of Fitzpatrick. Their ability to sunburn and their ability to tan is included in this assessment. When we were assessing the global markers that can be used for prediction, we found that in our assessment of 77 markers, we looked at the phenotypes and we found that there was actually a better accumulation of the type 3 and type 4 as an intermediate category because they were so close. And so we condensed the 6-scale Fitzpatrick scale into a 5-point scale used for our global prediction. So rather than having the 6 types of Fitzpatrick scale variation, which you can see here, we actually produced a new scale of very pale, pale, intermediate, dark, and dark to black. We then assessed within our individuals and our markers, well, what were the best or the most concise set of markers that could really give us a high probability prediction. And from AIC calculations, we found that 36 markers from 16 genes were the most beneficial towards global skin color prediction. Using multinomial logistic regression, we then formed a model based on these 36 variants on an assessment of over 1,400 individuals from over 30 populations around the world. In our assessment, we tried to include 1,000 cross-validations, so that would be splitting in our 1,400 individuals into an 80% training set and a 20% test set, and doing this 1,000 times to assess our degree of error. And as you can see, if we split our categories of very pale, pale, and intermediate, which would be deemed the white category from the snipper tool, we still have a high degree of information. They're a little bit lower than dark prediction and dark to black prediction. However, we tend to look at 2 categories as our most probable prediction, and I'll talk about that in the next slide. But AUC calculations here show that using these 36 markers for skin color prediction gives you a high degree of accuracy, especially if you want to look at white, black, and white, dark, and dark to black. Obviously, with Europe and with the variation that is involved in Europe, there is a big difference between someone from Northern Europe versus someone from Southern Europe. So that is why we stuck with our 5 categories and try to infer the difference between someone who is very pale and someone who's intermediate, because this is an important factor, especially for forensic investigators, because we can say that at a certain time of year, if it is in the summer when there is a propensity for tanning, someone who is predicted as very pale is highly unlikely to tan. And so you can give a prediction that is, I'm looking for a very pale individual, and that will not change from winter to summer. However, if you are looking at a prediction that gives an intermediate predictive result, then you have to factor in that if it's winter time, it is unlikely that this individual is going to change their skin color. They will be quite pale. However, they have the propensity to tan in the summer months. So you need to look for someone who is slightly darker or has a tan. And so having that factor included in the deemed white category was very important for us. We also did a direct comparison with the snipper tool because we wanted to show, okay, well, how good are both tools? How are they performing? Which is essential in assessing models and prediction outcomes, because we want to make sure that you as users have the most information possible. So from this prediction model from the human genetics paper of this year, we had a separate set of 194 individuals where we did have quantitative measurement. So we followed the role of snipper, their tool and their paper, and we ranked them the exact same way with regards being white, intermediate and black, using quantitative measures by snipper cohort. We then assessed how accurate were predictions from the 36 plex Harris plex S versus snipper's 10 plex. And you can see that there is a comparative result for both white and intermediate, which is expected because the accuracy of snipper tool was quite good for white as well as Harris plex S. But the black component is where there was a very high degree of improvement using the more global 36 Harris plex S. So our current research is actually bringing all of this together and yet again making a tool for forensic practitioners. We want to make it as easy as possible to generate the genotypes needed for imputation into our online tool. And so the Harris plex system, Harris plex S system is in the works. We've actually submitted to FSI Genetics and we're hoping for a good review of our paper there, and we hope to be published quite soon with the release of this system. This paper has several of the next slides, so you're seeing it before others. And basically, the Harris plex S system is an amalgamation of all three models. So you have the Iris plex eye color model, the Harris plex hair color model, and now the new additional skin color model of 36 SNPs from 16 genes, termed the Harris plex S model. That is involved in the prediction. And on the other side, the more practical side of the generation of genotypes, you have the two assays, the original Harris plex assay of 24 markers, so that's 22 variants for hair color, 6 variants for skin color, or for eye color, excuse me. And then you have an additional HPS assay consisting of 17 markers. With the technology of snapshot and for running on the capillary electrophoresis, we are limited with the number of SNPs that we actually run at one time. That is the reason for splitting this assay into two. With technology, as you know, and perhaps the movement towards next generation sequencing and massive parallel sequencing, we won't have to do this. And so there may be just one assay that actually gives you the full pigmentation profile. And that's certainly something myself and Manfred are working on. So we have 5 categories output from the skin color tool, as you can see there on at the bottom of the screen. And this is the picture of the second assay, the Harris plex S portion of 17 plex. As you can see, we can achieve accuracy yet again down to 63 picogram input. So if you were involved in the lab, you had your DNA profile, it was a low, or a DNA sample, it was a low STR profile, you needed extra information or some form of intelligence information, you can run simply 2 assays. It will involve only 1 microliter per assay of very, very limited portions of DNA. So you can talk about running a minimum of 60 picograms DNA input to achieve the 41 markers that you need to generate an eye, hair and skin color tool prediction result. This is kind of an example of where we are at right now with the skin color portion of the tool. I'll just zoom in on one individual. The 5 categories are given. They all sum up to 1. The highest probability is our first indicator of what skin color the person is going to be. But it is also important to factor in the second category of the prediction result. And for this, I'm going to actually go on to the next slide. What we wanted to do when we created the model was completely take out the ancestry effect. We did not want to have dependence on ancestry to infer a color prediction for skin. We wanted to make sure that we had the best markers available for global skin color prediction. And for this slide, I wanted to show you a couple of individuals from around the world and the number that's associated with their skin color and where they lie in a prediction, and how our global skin color prediction model actually performed on that individual without knowing anything about that individual. It was solely on DNA, irrelevant of ancestry. And so from this individual, their birth country was in the US. We were able to predict that this person had very pale skin, and so their category would be very pale to pale, as their highest probability was very pale at 0.66, but you have to factor in the second category as a contributor to that first category. So in this instance, we would say that this person has very pale to pale skin. For this individual, which is from number 7, this individual is actually from China. We wanted to make sure that our model incorporated both theories of evolution. So skin color evolved within Europe, but it is also believed that there was convergent evolution with skin color within Asia. And we wanted to make sure that the markers that consisted in our skin color prediction model factored this in. And so we wanted to differentiate that an individual from Asia would have lighter skin color, yet they would have black hair and dark brown eye color. To differentiate an individual from Africa who would have dark to black skin color with black hair and dark brown eye color. So this individual here was predicted as intermediate, so they have the ability to tan, but they are quite pale in their complexion. This individual here, number 11, is from Mexico, and their prediction here shows that they are darkly skinned, and it is such a high prediction that it is the most probable prediction is dark. And this individual here is actually predicted as being dark to black, with their high prediction probability of 0.82. And these individuals' parents are actually from Vietnam. So as I said, we wanted to take out the element of ancestry. We just wanted to focus on what the actual pigmentation palette of that individual was. Skin color prediction worldwide, yet again, we like to take out the same HDP set. We want to assess how do our markers perform, how does our model perform. Obviously, there is a lot more variation when it comes to skin color around the world, but based on these markers, we could infer that it does work reliably independent of genetic ancestry to the best of our knowledge, as we don't have phenotype information from this set. But you can see that there is a general idea that there are more paler individuals within Europe, and there are more darker individuals outside of Europe. So interpreting the result, as I talked about a while ago, it is very important to factor in the 2 highest probabilities when it comes to skin color prediction, because they can have a differing effect. You may not find a huge difference in these individuals from A and B, but I do, especially when it comes to the tanning ability, as I discussed earlier. For the top individual, they would be predicted as having a very pale to pale prediction. So because their pale prediction category is 0.58 and their very pale is 0.38. So that combination here means that this individual would be pale to very pale. Yet at the same time, individual B has the same P value or the probability value for pale. However, they have a higher value for the intermediate or the more tanning ability. So although this individual may look similar with the naked eye to individual A, if you factor in summertime when there's sun and when there's tanning ability, this individual B will appear darker than individual A, who will not be able to tan in the summer months. So that's why we like to factor in both the highest probability and the second highest probability category. Another example here, a dark individual predicted at 0.69. Yes, this is the prediction of the individual. You can see the color of the individual, and they have an intermediate category. So they are a little bit lighter than the dark prediction category. You have to factor in that this person may appear intermediate to dark. The individual at the bottom in D has the same probability for the highest probability prediction category. However, they do have a higher proportion of dark to black. So this individual will be darker than individual C. So having this in your interpretation is very important. The highest probability being the main category, with the second probability prediction influencing that highest probability, especially in the summer months. So now we're just going to do a few examples of the HBS online tool, because I do want to show you how easy it is to generate these probability results. You're all practitioners, you're all biologists, and some are interested in DNA profiling. So you're well capable of generating the genotypes involved in these assays. As I said, the HBS tool, Harrisplex S tool, consists of 41 genotypes that you need to generate. You can run 2 assays to generate these genotypes. And in the future, I believe that you can run 1 MPS assay to actually generate these results as well. And then all you need to do is simply hop online to access the online tool for prediction, Harrisplex S tool. And as I said, we continuously try to improve this tool so it's not stagnant in its predictions. The more information, the more data we generate, we try to improve prediction overall for eye, hair, and skin color. So if we just start with example 1, this individual was self-classified skin type 4, which is dark. This individual actually could be skin type 4 to 5. They're quite dark. Hair is dark brown, eye is dark brown. What I tried to do is show you the website and how easy it is. Right now, it's not online fully because we are still in the paper submission phase and the reviewers are assessing our paper. But I did want to show you, if successful in our paper submission, that this is what the website update will look like. The Harrisplex for eye and hair color is online, okay, but the Harrisplex S tool is not updated yet until the paper is published. So you're getting a sneak peek right now. Basically, it gives you a little bit of information about Manfred's group and my group. It gives you a manual for instruction if you need interpretation or how to interpretate eye color prediction, the highest probability, hair color prediction using the guide, and lastly, skin color prediction using the first 2 highest probability categories. In the Harrisplex S system, as you can see, are 41 variants, and we have it listed as the gene, the SNP name, the allele input that you must generate, and the number of alleles. So we choose based on our training data and our large data set, which allele you have to input. So we term it the input allele. For example, if you have at the first SNP of the first assay of HP, if you see a CA at that marker, then that is a heterozygote. And then you would have to put in that you've observed one of the A alleles. So you would have to type in 1. If you do not observe the A allele at all at that marker, and you simply see at that genotype that it is CC, you leave the number of alleles of 0. If you observe a homozygote A at that marker, then you input 2. If, unfortunately enough, you have a highly degraded sample that is even lower than the 60 picogram input, or it's highly degraded and you cannot generate that marker, you can still generate probability results. However, they will infer an error, so an AUC, a loss in accuracy prediction. But you still can get the majority of probability predictions for the categories. So you can input an NA if you do not have a result for that genotype. There are some restrictions into which genotypes you cannot afford to lose. And in eye color, you cannot afford to lose HERC2. And there are several other markers that you cannot lose for hair color as well. So as I said, you come down to the model. We include the number of individuals in our database. As you can see, there's a large number of individuals within our database for the different traits. And you can also see the current database accuracy levels at a population level. So how good are we at predicting a certain trait and the different categories that are associated with that certain trait? As an example, here, because it would take a very long time for me to input each of the individual alleles into many of the genes, we have this file upload version. This is great because you can have a whole list of individuals, all of their genotypes. It can simply look like this. And you have a sample ID of all your individuals, all of the markers from 1 to 41, and basically the presence or absence of the input allele. And from here, you can actually choose to upload this file and generate your probabilities. So it's a very, very easy to use system. When I generated the probabilities, it gives it some time, and then it automatically downloads your results. And if I was to open up those results, this is what you can see. You can see that there are 3 individuals. All of the genotypes here are given, just like the original input model or input file, but also you generate eye color probabilities, whether you had the full number of genotypes there or you were missing some markers, the hair color probabilities, yet again, were you missing any markers, the shade of the hair probabilities, and lastly, the skin color prediction probabilities. And I just have one example for the slides. As I said here, the individual was classified here as having skin type dark with dark brown hair and dark brown eyes. And if I look here at a tidied up Excel file of the exact same result, you can see that there are probabilities for brown eye at 99%, probabilities for brown and black hair. So this individual would have dark brown to black hair with this probability prediction using the guide, and this individual would have dark to black skin. Okay, so that's just some of the examples for prediction that we can do with HiresPlex S, and I'll just come back to the main screen. Okay, have I lost?
Dr. Walsh, since you were sharing screens,
Okay.
it looks like your screen share might have jumped off. If you want to go ahead and initialize that again, then you can just bring your PowerPoint back up.
Okay, so as I said, the capabilities of the HPS Online tool is very easy. Generating the genotypes is really easy with the Swignum validated assays, and it really is a very, very useful intelligence tool to generate some new avenues of investigation for investigators and for practitioners such as yourself. And from there, I can take questions.
All right, thank you so much. So we do have a couple of questions that came in so far. The first one was from Daniela. She asked, do you know what, one second, do you know what prediction model Forensic uses, and have you done any comparisons on their accuracy?
Do I know which model the forensic community uses? Is that it?
No, she's talking about the forensic assay from
forensic.
the
Sorry.
Yeah.
Yes, forensic assay actually uses the HiresPlex SNP variants, and so they actually generate pretty much exactly what we do with our online web tool. However, they have the older version of the prediction probabilities. So they don't have the capacity to improve the prediction probabilities like we do, because we generate new data all the time. So they're using the prediction model from 2014. So they do have the 24 variants that are required for eye and hair color prediction. And from that, you can input into our online tool and generate the most up-to-date probabilities for eye and hair color prediction.
All right. Next question we have is from Randall Libby. It's asking, what is the minimum SNPs which can be reported in Heuris-Plex to produce predictive value?
I can just see the question there. I'm sorry, could you repeat that? Oh, what is the minimum?
What is the minimum?
Okay. So what
yes,
is the
ma'am.
minimum number? What is the minimum number of markers, is it?
It's the minimum
Yes,
SNPs which can be reported.
Well, it depends on your genotyping quality. If you have run the assay according to the guidelines and our publications, you should be able to generate all of the markers required. And when you generate all 24, you will get the most probable predictions for eye and hair. That's for Heuris-Plex. It's the same for Heuris-Plex S, so that would be generating 41 genotypes for eye, hair, and skin color prediction, and that will give you the most accurate probabilities from our data sets. But there are certain factors, especially with anthropological and ancient remains, where you simply cannot generate all those markers. And on our website, and it's built into the online tool, if you are missing HERC2's RS129, you will not be able to predict an eye color result. That is because it is too important a marker for prediction, and there is no point in predicting without that marker. The same for hair color. There are certain combinations of SNPs, HERC2 being one of them, SLC being another, and OCA2 being another for hair color, where you have to have these available. All of this is on our tool. You can find it in the PDF guide on the online web tool. So it basically has all your answers there. But the tool actually itself will produce a 0 probability if you do not have the minimum SNPs for that trait.
All right. Chuck Hardy wrote in stating that he may have missed this during one of the talks, but he asked if hair type had been addressed, such as straight versus curled hair.
Yes, we're actually doing research on that right now, because obviously that would enhance our hair color prediction as well, having that physical trait included in prediction. Research is ongoing, and we are certainly looking at straight versus curly hair.
All right. Next question we have is asking, could you recommend any commercial labs who provide this service?
Well, I think that the question really is are you capable of running the assays yourself in your own lab? Because we have them developmentally validated. So in essence, they're quite like STR kits. You just need to run internal validations to generate the genotypes required. So it is a really easy assay to run. You have all of the equipment in your lab. So essentially you could set up a standard operating procedure to run these assays in your own lab. However, if you had to choose to not run it in your own lab, one of the users pointed out that yes, Illumina generate the 24 markers in one of their assays, which is a kit that you can buy, and also AB, Thermo Fisher, generate the same markers. So whether you choose to generate the genotypes yourself or whether you choose to buy a kit to generate the genotypes, it doesn't matter. Really, it's just generating the genotypes and using the prediction models to infer the probabilities. We just want to see these prediction models being used. We want the research to start being used because there are so many cases where DNA is in wonderful copious amounts. There are no matches in databases or suspects. Why not run these tools? Why not get extra information and give to your police officers so that they can go out and say, okay, we're looking for someone that possibly has blonde hair, blue eyes, and very pale complexion, because it may make the difference in your case.
Let me jump in for a second, Josh, because I want to follow up on this. Of course, Susan, you published a paper on the Hersplex developmental validation in, I think it was 2014. And so the tool is out there in terms of the technical aspects to implement it. But do you know of any laboratory that has actually done an internal validation of the assay? Because I think many, if I think as a practitioner here in the U.S., many are thinking, what would it take to do an internal validation? What kind of resources in terms of time and number of samples to run? Are there any standards? So that's how people, I think, are thinking about doing internal validation. So I was wondering if you had any insight on that.
Yeah, absolutely. So the same types of methods that are involved with STR typing and the new kits that you use, especially global filers, come on board now. So there are a standard set of guidelines on internal validation. So you would have to run several individuals running on your machines with your equipment. There are lots of standard operating procedures. Because we are using the same equipment, the exact same equipment as an STR kit, the only difference being that it's snapshot chemistry and purchased primers versus a kit that you buy in a box. It is very easy to assign a person that can do a validation within a few weeks to the high levels of any of the purchased kits. And this is very easy to do. We are here to help. I myself actually run some samples in my own lab on cold cases for Indiana State Police. So we are working together to generate some intelligence information on cold cases within the state of Indiana. I am happy to run a casework for individuals, or at least to help them run it in their own lab so I can assist in training. But at the end of the day, technology is moving that Illumina and Thermo Fisher are actually generating these kits for you. So I would worry less so much about how to generate the genotypes because that will come in time, especially with the advance in technology. But I am here to help. If you do want to start running these assays, I'm only an email away.
All right. We've got just a few questions, and then with the interest of time, we'll move on to our closing. So just a clarifying question. The prediction model that you used, that is based on the Bayesian model, is that correct?
No. The prediction model that Hyrspex uses is based on multinomial logistic regression. So it's a regression model, but using input of 1,000 individuals and an export of probability prediction. That is a probability value. So that's the percentage that I told you, whether someone is 90% blue-eyed or whether someone is 50% blue-eyed. With the Bayesian model, that is the snipper model. So that Bayesian approach basically conforms the probabilities into likelihood ratio, which is the person is 100 times or 10 times more likely to be brown than blue. And so we feel that using the probability output is more beneficial because you can see how the genotypes are contributing towards each category, especially important in hair, understanding hair prediction results and skin color prediction results. We find that that's lost in likelihood ratio. So although likelihood ratio is preferred in STR profiling and in court, and some practitioners may prefer likelihood ratio, I as a researcher and Manfred, I may speak for him, probability prediction output for the categories tends to give us more information on the genetic side of prediction.
All right. Next question we have comes from Holly Porter. She's asking, or she states that several people have told me that their eye color has actually changed over time. Does eye color change, and does eye color change, and does that correlate with hair color change as well?
It is actually very rare for eye color to actually change. Probably what the individuals that are suggesting that they did have eye color changes, maybe they had an injury as a child or an individual where their eye actually, it's actually seen as brown. It could be coagulated blood, which is actually the case of David Bowie, for instance. One of his eyes is different because of an injury to the eye. But eye color in general does not change in one's lifetime. The melanocytes and the melanin that's produced within your eye only occurs really at the early stages of infancy. And you do not regenerate these like you do hair and skin. The only difference that you're possibly seeing in eye color is uh, whether you're inside or outside and what you're wearing, and because it's actually the scattering of light from the eye reflectance that gives us the color of eye. So technically your eye color isn't changing, it is just the perception of your eye color that is changing. The only way that eye color changes is in old age. And all that's happening there is the decrease in intensity of color. If you've ever observed an older person, you'll see that they have a kind of a film of opaque film around their eye color. So it just seems less intense in color. But it does not actually change the genetics. The genetics does not change, and the proportion of color in there is the same. It just looks a little duller.
Okay, we've got two questions left. The last one, or second to last one, is from Randall Libby. He's asking, are you aware of any labs or individuals that have attempted to introduce this type of data into a criminal court proceeding?
I believe that the only way that this can be introduced into a court proceeding is if it would undergo a Fry hearing, I believe, in the United States, and that would just be testing the reliability of the reliability of the technology, and that would need to be that would need to occur in the United States for this to actually come into court, because it's an intelligence tool. The only information really that is needed is that the police investigators did their job, that they went out and they did an assessment of the crime, they did their research on eyewitnesses, they did their research on the use of DNA, and that they found a most probable profile. And from there, they apprehended an individual and then they took their STR profile. So the only way that this will actually be in court is that the police would say, we had some DNA intelligence information that led us to this individual. But the actual genotypes and the probability predictions, it is unlikely that they will be talked about in court other than a Fry hearing, which would show that the technology is sound, that the genetics is sound.
Okay, and our last question for today is from Beth Harris. She states a lot of profiles that we get in forensics are mixed. She's asking, or she's assuming that this may not work as well with mixed samples. Is that true?
Yes, with the current technology and SNPs, it is very difficult to differentiate mixtures. Right now, we have a couple of mixtures, but that we can obviously separate out with more technology methods, such as differential extraction. We can separate out mixtures that way, and then proceed with singular profile generation. We are hoping that with the research that myself and Manfred are currently doing, that we are going to try to see if mixture deconvolution can occur with next generation sequencing, and that is possibly the biggest avenue of research that next generation sequencing is going to bring to the table, to see if we can deconvolute mixtures. And obviously, these types of assays would fall under that category. So yes, we are working on it. Uh, Obviously, in intelligence information, the STR profile is the first thing that's generated in a standard lab, in a standard DNA profile lab. For intelligence information, we will go on that STR profile. So ours is not the first tool that's used, and that's something that's very important to know. We are the second tool that is used. First, an STR profile is generated, and from that STR profile, you will be able to see whether it is a mixture or whether it is not a mixture. And from there, we encourage people to use single profiles to generate this intelligence information. It is more difficult and it is impossible to separate out mixtures on the current CE platform. But I do believe that we may have encouraging results using MPS or massive parallel sequencing to differentiate mixtures in the future.
All right, thank you so much, Dr. Walsh. We definitely appreciate your time and your presentation today. We are going to go ahead and move into our closing. Daniela, do you have anything that you would like to leave our audience with, such as upcoming webinars?
Yes, thank you again, Susan, for your presentation. That was really interesting and full of a lot of information. So during the presentation, Dr. Kaiser talked about a webinar that's scheduled in the future about predicting the age of an individual, and I wanted to say that that webinar is scheduled to be on the 6th of December at the same time. And in a couple of weeks, specifically on November 7th, we will have a webinar on the history of SNPs as a forensic marker. And so if you're interested in learning more about SNPs for, in this case, for individual identification, then please join us on November 7th. And with that, I think, Josh, you can close.
All right. Thank you so much, Daniele, and thank you so much, Dr. Walsh. We definitely appreciate your presentation. Thank you so much, Dr. Kaiser, as well. So much great information was given out to our audience today. We would obviously also like to thank our audience for attending. Without your attendance, we definitely would not be able to put these webinars on, so your attendance and support is greatly appreciated. You will see there in the middle of your screen, we do have a link to our short customer satisfaction survey. Your feedback for the webinars is vital information that we always use to help provide you with the best experience and to adjust the content and also the way that we may disseminate the information out to you. So please take a couple of minutes and fill out that and provide your feedback. We would definitely appreciate that. As Daniele said, we also have some upcoming SNP webinars, one at the beginning of November, November 7th, and then one December 6th. So be sure to get registered for those once we push those out to you. Again, thank you everyone for attending today. Thank you for all of our presenters for taking the time out to deliver your information. Make sure you complete today's survey and also check us out at our website, www.forensiccoe.org. I went ahead and pushed a new web browser out to everyone that's still in the room. So that should open up to you there. You can find all of our upcoming courses as well as everything that we have done previously. It's all been archived and recorded for you to view at any time. Also follow us on all of our social media pages, Facebook, LinkedIn, and Twitter. We're constantly putting new content in there. Again, we would like to thank everyone, and we hope everyone has a great rest of their day, and we look forward to seeing you in the near future for our future webinars. Have a great day, everyone.